of different computational techniques to evaluate the predictability of spillover events from wild reservoir populations to humans. That's a mouthful, but obviously it is very relevant to what we're talking about today. So I want to give you all a few reminders before we start the discussion. Remember that if you're on Facebook, you should search for the ICFJ Global Health Crisis Reporting Forum group and join it there on Facebook. We are running forums in 5 languages now: Arabic, English, French, Portuguese, and Spanish. And we have more than 10,000 journalists around the world participating in the forums. So if you have not already joined the forum, please look for it there and join us. If you're not on Facebook, you can visit the COVID-19 resource page on www.icfj.org, and there you can sign up to receive our weekly newsletter, in which we'll give you news and links on upcoming webinars. So even if you're not on Facebook, you'll have an easy way to keep up with what we're doing on the forum and join all of our future webinars. You should also visit ICFJ's publication, ijnet.org, which is the go-to place for stories on the latest journalism trends, tip sheets on just about every practical aspect of journalism, and opportunities for training and career advancement for journalists. And we do that in 8 languages on IJNet. Now, I also want to remind you that you can please type in your questions during the discussion in the Zoom chat on the icon below. You can also post your questions on Facebook if you're watching us there. Right below the video post, you will see a place to post your questions, and my colleagues will then post those questions in our chat here. So I'll be able to ask Bernardo Gutiérrez your questions. So today's topic, the UK variant and the similar South Africa variant that we've been reading quite a bit about. These variants have arisen in their respective countries and have now spread throughout the world. They are much more transmissible than the coronavirus that we've been dealing with for the past year. And they are causing great concern, therefore, just as we are beginning to roll out vaccines. Some experts have said that we are now in a race against time to get people vaccinated, as cases are exploding in many countries, in part because of these new variants. Much of the UK, where Bernardo Gutiérrez lives, is now on an extremely strict lockdown, with as many as 1 in 50 people in England and 1 in 30 in London infected with the virus. Some people have started calling Great Britain Plague Island. A little bit of maybe hyperbole there, but gives you an idea of how serious this is in the UK. So what we want to do now is explore how much the rest of us around the world should be concerned about these new variants. Bernardo, we're going to be talking about the science behind the UK variant shortly, but first I'd like to ask the most important question. What should we be worried about with this variant, and what shouldn't we be worried about?
Well, first, thank you very much for the invitation to participate in this webinar. It's definitely a good question. And my go-to reply to this would always be, of course, it's not about being worried, it's about being vigilant. So it's just something that we've learned over the course of the last year, that if we have the ability to have a good and efficient way to tracking cases and getting as much information of those cases as possible, this includes, of course, genetic sequences, we can gain the upper hand with the virus. Now, for the specifics, of course, what should we be worried about? Based on what we see so far.
Apologies, I call her the Secretary of Homeland Security.
Yeah, doing her job, looks.
Right.
So yes, so the one thing that we're definitely looking at the hardest at the moment is the possibility of the virus being more transmissible. So that means that for every person that gets infected, that more additional cases can come out of this original person that carries the virus. What we shouldn't be worried about, at least based on what we've seen at the moment, is that this new lineages may be particularly keen on escaping the immunity that you can produce by getting a vaccine, or that the type of disease that's produced is any different. So as far as having variants that are potentially more lethal, for example, that's not something that we're looking at from the data at the moment. But of course, I do want to go back to my original message. This is about staying vigilant and keeping track of things as they develop, because I think for all of us, the last year has been one big learning experience about how quickly the situation can change and how we need to adapt to it as it transgresses. So that's something that's important, just keeping that spirit of not necessarily fear, but more about, okay, let's keep our eyes open and try to be as vigilant as possible and trying to actually invest the resources and the time to engage in this kind of activities.
Okay, thank you. Now, what I've read is that this variant in the UK, at least, is said to be about 50% more transmissible than the virus that we previously were dealing with, or the virus variant that we've been dealing with. What exactly does that mean when we say it's 50% more transmissible?
Well, so first of all, the number 50% is basically the mean from different estimates and different levels of confidence that we have on our estimation of how much more transmissible it is. So there's been different reports written using different analytical approaches. And from there, we've got numbers that range from 30% more transmissible to 70% more transmissible. But there is uncertainty to these numbers. So the overall mean seems to be somewhere around 50%. Now, what does that mean? Based on the kind of analysis that have been done so far, basically this means that if you compare this particular lineage in the UK, and if you compare it against every other lineage that's currently circulating in the UK, you tend to see that the number of new cases increases roughly about 50% faster than others. Now, of course, this is based on this very specific setting. Whether this means, for example, that if you go out on the streets, if you personally are 50% more likely to be infected, that's something that doesn't necessarily translate that directly. So there are going to be numerous factors that we're definitely working on trying to understand. And by we, of course, I mean we as in the scientific community. But the full range of implications of this change in transmissibility, that's something that we are working on at the moment.
And I've read that the variant has been discovered in at least 45 countries by now, but it's likely present in many more than that, right? Can you explain why many countries may not have even discovered it yet?
Well, this actually could be taken back to the beginning of the pandemic. So when the virus began to spread outside of China into different countries, there were a lot of countries that, just because of how their surveillance systems were set up, so whether the different public health agencies were looking for cases, they didn't have either the capacity or the technology or the resources to identify cases early. So something that we've seen with this virus in particular, and to be honest, it might be something that happens with viruses that transmit in similar ways, is that the virus transmits quite rapidly and associated with human mobility. So whenever you have people traveling between different destinations, these people tend to be the main carriers of the virus from one location to the other. And in a few countries, we've seen that by the time we're starting to detect cases, that means that there is some degree of circulation within the communities of that country. So a typical scenario would be, for example, if you're starting to get the first cases, normally it's quite common for these to be travelers that are returning from another location where the virus was already circulating. But by that point, there may be some cases that were missed, and those particular cases might have already transmitted some people. So it's something that we've seen over and over again, and it's not rare for countries to pick up the first cases at a point at which they can't really certify that every single new case is from a person who's returning from another country. So this probably has to do with, again, numerous factors. And at the end of the day, the result of this is that whenever you're starting to detect cases, it might be that the virus is already circulating. Now, a little addition to this is that given that to detect these specific variants, you need to do a lot of genomic sequencing, this puts an additional pressure because it's not about detecting cases and looking at symptoms, which tend to be a bit more straightforward. this is more of a situation in which to identify new variants, you need to be doing genomic sequencing. And that's something that is still quite irregular across the world. There are countries that are doing very good job and they're bringing a lot of resources and time and expertise to it. And there are countries who are still building up their genomic surveillance. So that is going to be a factor that partially determines whether any country will detect the variant early or it's going to take a bit longer for them to figure out that the variant is already present.
And as I understand it, the UK has long been very advanced in genomic sequencing. They do a lot of it. United States, where I live, is not so good. So even here, we really have no idea how widespread this variant is because we're just not doing the level of genomic sequencing that the UK is doing. Is that
yeah. And I actually see that for a country like the US, it's going to be definitely challenging because it's a very large territory and there are a lot of differences between individual states and how they handle their surveillance, their epidemiological data. So to having like a single strategy to perform genomic surveillance can be more challenging in some countries than others. As far as the experience in the UK, it was definitely an initiative that started from a proposal from big funding agencies like the Wellcome Trust. It had the support of government agencies and government epidemiological surveillance. So for example, Public Health England, Public Health Scotland, they were involved from the early stages in this process, and the academia, because academic institutions had considerable expertise, they had a lot of technical knowledge, they had resources that they could also contribute to. So the strategy in the UK has definitely been by far one of the more developed as far as sequencing as many not as many variants, as many samples as possible, which has, of course, a lot of results, but this was a product of a very decisive early decision on actually like investing and coordinating the work. Because end of the day, the number of sequences that we're seeing in the UK is the product of lots and lots and lots of small agencies, small universities, small testing centers doing their sequencing in a very decentralized way, and then a big pull to share the data. So I think that's been something that clearly played a big role in how successful it's been. But for other countries, there's going to be different approaches, and sometimes they're more successful, and sometimes they present definitely more challenges.
And tell us a little bit about the South Africa variant, which is a different variant entirely from the UK one, but also seems to be highly transmissible.
Yes, so the South Africa variant, which, well, all of these variants, they do have their specific names, which are a bit hard to follow. So the UK variant is B.1.1.7, South Africa is B.1.351. But in any case, for simplicity, the fact that they have these different names comes from the underlying fact that they emerged in completely independent events. So as the virus has spread all over the world and the virus starts to just transmit in a country that perhaps has very low number of travelers that are connecting with other countries, the virus that is just circulating within the local population is going to start to accumulate mutations and change a bit. And so the probability that different variants emerge is something that's just been very common all over the world. No, so the first thing is precisely that. The South African variant has a very different origin from the UK variant. The other point is that some of the mutations that drew the attention of researchers early on are present in the South African variant, but not all of them. So the key mutations that we're looking at are actually slightly different. The South African variant has two particular mutations in a very important part of the virus that potentially could be associated with how it's becoming more transmissible. The UK variant has one of those mutations, which might also play a factor. So this might also have something to do with whether the virus transmits more or less. Once again, just something that we're looking into at the moment. But they have different, end of the day, they do have different mutations, and they can be behaving similar to a certain extent, but they are different versions of the virus, so to speak.
And just yesterday, Dr. Anthony Fauci, the director of the National Institute of Allergy and Infectious Diseases here in the US, said that we may have our own mutant in the US that is more easily transmissible. He said, quote, "We don't know, we're looking for it. If you look at the slope of our curve, which is very steep, it looks a bit like the curve in the UK. So could we see even more of these highly transmissible variants arise around the world?"
Well, yes. Yes, but there is a catch to my answer. So first point is, this is not something new. New variants have been emerging as the virus spread in different places all over the world. The thing that we're starting to look at the moment is that these variants may be transmitting at a higher rate than others. So most of the variants that have sort of just emerged as the virus transmitted so far, they tend to transmit roughly the same. Actually, early in the pandemic, there was another mutation that drew the attention of researchers. It was on the same protein where the current mutations that draw attention are located. And it seemed to be that it did transmit a little bit faster than other variants. However, in the bigger picture, it didn't quite affect the total number of cases and how the virus spread at the end of the day. But of course, as time progresses, the chance of new mutations happening just increases. So the fact that there may be new variants with mutations that affect how the virus behaves, it is definitely a possibility. And not only that, but actually over the last 48 hours, I would say, there have been a few countries reporting lineages that carry specific mutations. So at this point, it's probably worth knowing that whenever a country reports a variant that may be more transmissible, I would think that the most likely sort of mindset by which they determined that it was potentially more transmissible is that they identified a whole cluster of sequences, a whole cluster of cases, and they all carry one of these mutations that are noteworthy. So it doesn't mean that they necessarily know that it's more transmissible, but they carry these markers on the genomic sequence that may suggest that that's what's happening. So that's one of those things. Now, the other thing, and maybe, and I'm definitely not as familiar with the statement that you mentioned by Dr. Fauci, but it's still hard to determine whether the increase in cases is directly caused by a new variant. So for example, in the United Kingdom, definitely, so before the current lockdown, there was a 1-month lockdown that happened. It was right before the Christmas holidays. So it was in November and early December. During this time, we did see the cases started to decline, but the decline wasn't necessarily like a complete squashing of the curve, as has become a common term. But it actually, the decline was sort of happened right at the time at which the lockdown was lifted. And after that, you saw the increment in cases. So there are different factors that could play a role here. It could be that a new variant is really contributing to additional number of cases, but it could also be, and based on our understanding, at least at the moment, there are lots of other factors that also contribute to increments in number of cases. So the fact that there was still some virus circulating when the lockdown was lifted may have played a role as well. So it's important to not necessarily interpret an increment in number of cases as the product of a new variant. Actually, whether this variant heavily contributes at the moment to the increase in number of cases is something that we're studying, because this is something that it's quite important when you're doing research, and it's perhaps something that's even become quite common to hear people say, correlation doesn't equate causation. So mostly we identify things and we say the increment in number of cases happens at the same time as this new mutation. But to actually try to establish whether the mutation is causing the increment of cases, that's something that takes a little bit more work, and it takes hopefully more lines of evidence. So not only this observation of we see more cases and we see a mutation, but actually taking the virus to the lab, looking at those mutations, seeing what they do. So end of the day, and the reason why this takes a little bit longer than people would like to draw really robust conclusions is because it's a long process, and we usually expect to have very different lines of evidence that reach the same conclusion. That's when we're we start to become more certain about this causation. So whether something like a mutation causes an increment of cases.
And that's the research you and your colleagues at Oxford are doing right now, correct?
So yes,
this
actually,
is genetic.
Yes, the research that's being done for this variant. Well, first of all, it's definitely worth noting that while I'm not personally involved, but I'm part of a research group that has a few people that are working on it. And secondly, it's not only here in Oxford, but it has been, same as I was talking about the collaboration to actually do the massive sequencing in the UK. There's lots of different groups working on different questions, on different approaches to answer the question. So there are groups in various universities and various public health institutions that are actively working on this. And there are going to be different approaches. So from my side, it's going to be about looking at the sequences and trying to figure out whether there are associations that are predictable and that convey the same conclusion as a model in which the mutation causes the increment of cases. But at the same time, there are other groups, for example, in different universities and in Public Health England, and they're working at actually taking the virus to the lab, looking at those mutations, isolating viruses with that particular mutation and seeing whether these viruses behave differently when they infect cells in the lab or when they infect animal model in the lab. So it's different lines of evidence, again, that we're really seeking here.
Is there a certain time frame when you expect that we would know whether this is really a genetic variation that leads to higher transmissibility, or if it could be really just something circumstantial, as you said earlier, super spreader events or something like that? Do we know that yet, or is that confirmation still to come?
Well, I think it's an ongoing process, and rather than setting a date in which we say, okay, now we know for sure, I think our confidence is just going to increase on one answer or the other as time progresses. But having a time frame at which we can say, okay, this is certainly the right answer, it's hard to tell, and I probably don't have enough experience in the field to really tell that those timelines. I do know, for example, that the results from lab experiments, that's something that we're definitely looking forward to, and they may be available relatively soon. I think there is actually some preliminary reports already out in the public, which they still need to be verified, going through a review process. But yeah, unfortunately, I don't have a specific date when we might be certain. I just think it's going to be this. We're going to be more and more certain as we see evidence, and especially as we see different countries with their own local transmission of the variant. So it's unfortunate to think that different countries are going to have to deal with the virus from this variant or other variants that may be behaving differently. But the fact is that the more countries doing genomic surveillance start getting data out, that's also going to be very helpful, because if we see the same phenomena occurring in different independent places, that's going to be a strong signal for us that, okay, maybe this is something that happens again and again and again, and that's a good sign of an indication that there might be something worth looking into or supporting the conclusion.
Great. I want to remind everybody to please post your questions in the chat, and we will start taking those in just a few minutes. I see that some questions are already coming in, so we're glad to have all of your questions as well. Bernardo, for those of us who are not scientists, the word mutation sounds really scary. But viruses are mutating all the time, actually, and usually in ways that aren't harmful. So tell us more about how this happens, a little bit about the science of it, and what is it that makes the new variant so much more transmissible?
Well, no, and that's a great question. And I think one of the words that has been lingering more or less during this pandemic has been the word mutation. And yes, as you say, the viruses, they just naturally accumulate mutations. So basically, I guess the easiest way to explain this is, say I am infected with the virus and I transmit it to 3 other people. So that means that basically the cells that are infected in my body will produce the small virus particles, and they will eventually go through an aerosol and enter the other people's bodies and infect their own cells. in this entire process, each one of those cells that is going to infect a different cell, when the virus is making copies of itself, its full genetic code is going to have some mistakes as it just generates the copies. So that's a very natural process. It's actually how evolution moves forward. But as we start seeing these mutations, so say I transmitted to 3 people, those 3 people, one of them didn't transmit to anyone, but the other 2 transmitted to 3 people each, and then one of them transmitted to another 6 people. And so you can see how it starts to fan out. So these little mutations, little changes in the genome, as the virus spreads, you'll start to see as they accumulate. So if after this process you look at all of the people that were infected and you compare the genome sequences, you can actually see how there are changes that arose as the virus just went from person to person. So that's a very natural thing. It's something that we've seen with tons of other viruses. Actually, my entire PhD project is only possible because we can actually use these mutations to track where the virus has circulated. But given that this is a very natural process, the majority of the mutations are going to be either neutral or maybe even slightly non-beneficial for the virus. So the changes happen, but the results as how the virus behaves, what type of disease it causes, how fast it spreads, for the vast majority of situations is going to be just neutral in the end. Now, every now and then, there is a possibility, because the mutations happen at random on the genome, that one of these random changes will actually make the virus a little bit better at spreading. So in that case, if that mutation occurred when I was infecting 3 people, for example, if one of these first 3 people, that's where the mutation happened, there is a possibility that that person is going to infect to 10 people on the next round instead of just 3, as happened in the first round. And then each one of those 10 people is going to transmit to 10 people in turn. So there you can sort of get an idea of how one specific mutation, if it benefits the virus and makes it easier to transmit, it might make it to just become the more common genetic change of the virus as it circulates within a population. So that's basically what we're looking at. And of course, you do need to be very mindful about whether the 10 infections that happen because of this, when this mutation happened, whether they were caused by the mutation. So whether this change actually changed the way in which the virus behaves, or if this has something to do with the person that infected 10 people attended an event at which a lot of people were involved and were exposed. So there are these factors that always play a role. And I'm guessing the hardest thing to do is to actually work out how much each of the factors contributes. And that's probably what takes the longest to actually try and understand. So whether it's actually a genetic change causing this different behavior, or whether it's some other circumstance that's playing a role.
Right. And one of the things that has caused people to worry about in any mutation is whether it will cause more severe illness, lead to more death, that kind of thing. And as I understand it, there's no indication that this variant is more lethal or causes a worse case of COVID-19 than previous variants. But one of the stories I read said that a highly transmissible variant could actually lead to more deaths than a more lethal variant if we were ever to see one. And one way to explain it is that a small percentage of a very big number can easily be much, much bigger than a big percentage of a small number. So is that I think, do you think a valid way of looking at the danger of these kinds of variants?
Yes, especially when we're building possible scenarios, given how little we know about this new variants and any new variants. Yes, I think that is definitely something worth considering. So I usually like to just frame these things as in mind experiments. So as an example of a mind experiment, let's say that you take a copy of London. So it's the same as real London in every possible way. The only difference is that in one of the 2 versions of London, you have a virus that's more transmissible. And in the other one, you have a virus that is more lethal, but transmits the same as the rest. And you just think, okay, what would happen if you move forward in time and see the number of deaths associated to this. So of course, if you have a variant that is spreading a lot faster, and end of the day, that causes a lot more cases, the small percentage of fatalities from this outbreak that I'm describing could still be a lot larger than the alternative version of London, where perhaps the virus actually does kill 50% of the people that get infected, but is transmitted in such a limited way that there's going to be a few people who are unfortunately going to be in contact with this hypothetical version of the virus and die from it. But on the bigger picture, it won't reach as far as it won't transmit as far as to cause more deaths. So yes, that's definitely something worth considering, especially when just building scenarios. So one interesting way to look at it is, if the virus transmits at a faster rate, on the long term, could it overtake all of the other variants? Is actually the fact that the virus is circulating together with other variants at the moment, does that actually play a factor in how it will behave on the long run? Do we just expect it to keep on going and overtaking every other lineage that's present, or at some point there is some other force that counteracts this? Another interesting question is, at what point, so what percentage of the cases need to be associated to more transmissible variants for it to actually just drive the entire epidemic curve? So basically, at what point, how many of those cases from the total number of cases need to be from a more transmissible version of the virus for the entire number of cases from the entire pandemic to change from its expected trajectory? So yes, it's definitely something worth keeping in mind, and it could potentially have big implications, but yet again, it will really depend on a lot of factors, and our best tools are, end of the day, working with limited knowledge and information that just comes day by day. So it's definitely wise to always keep an eye open and ears open to the new information as it develops, but at the same time, be very careful about taking preliminary information as it is, as preliminary, and still being able to be further dissected by the scientific community or the public in general who might be looking at it.
All right, great. I'm going to ask you one more question, although I have lots more, but I'm going to ask you one more before we turn to audience questions. And that is something that's very important as we ramp up vaccinations around the world, although probably at a much slower rate than we had wanted. How concerned should we be that the new variants could make vaccinations less effective?
So, of course, following the theme with this webinar, the investigation is ongoing.
Okay.
But fortunately, the analysis that have been done at the moment suggests that we don't expect it to affect the efficacy of vaccines. Now, this is particularly for the UK variant, which is one that's being analyzed in greater detail. There is other work looking at, for example, the mutation from the South Africa variant, and it suggests that that may have a bigger effect on how the virus behaves, but it's something that we're looking into at the moment. But as far as the UK variant, from the data, it doesn't seem to make a major change that would make us think, okay, there is a possibility that it won't, that a current vaccine won't provide enough protection. Whether that will be the case and whether that conclusion will hold for this or other variants through time, again, as the ongoing nature of the situation, it's definitely something that we need to keep an eye open. But perhaps just to follow part of the rationale of why we're not that concerned at the moment is that whenever the body develops an immune response to a virus, it doesn't necessarily produce antibodies that only target a specific tiny part of the virus protein that coats the envelope of the virus particle. It actually develops different versions of antibodies that bind in slightly different ways across a protein. So if there's one mutation that changes one part of the protein, it is likely that the body developed other antibodies that bind to slightly different places in the protein. So that is part of the rationale, part of the constellation that we have at the moment, that one specific mutation might not play such a large effect in how the efficacy of the vaccine. But yet again, it's only time will tell whether this holds.
Okay, well, let's start taking questions from our audience. We have a lot of them here. And the first question is sort of gets to one of the questions I had, which is, about sort of what measures we should be taking as we learn more about these new variants. It's from Alex Hogan. Does a more transmissible virus make some of the standard protective measures, masks, 6 feet distancing, etc., less effective?
It's a great question. And at the end of the day, it is going to depend on what functional effects the mutation has. So in other words, whether a mutation changes the way the virus behaves in different stages of the transmission process. So if there were changes that were to make the virus more resilient to environmental conditions, for example, that may play a bigger role as to how different personal protection measures work. If the change only has to do with how the virus recognizes the cells of the body once it infects, then it might still be that there is a factor in which the same protection is not as efficient, because with personal protection, we do need to keep in mind that for an infection to actually generate a disease, it's not about a single virus particle making it through and getting to the lungs. You normally need like a minimum doses. And this minimum doses is the one that's actually going to cause a disease. So barriers in the end, they might be protecting from a large part of the potential inoculum, so to speak, the original sort of bundle of virus that enters the body. But perhaps even if the same number of little particles of the virus enters, one mutation can make these particles that entered with or without the personal protection more likely to actually get a hold of the cells and actually cause a disease. So that's probably like the long way to answer it. I wouldn't expect major changes at the moment because it is unlikely for a single mutation to have such a large effect. But if the change in how single viral particles actually affect the entire immune response and the entire mechanism of how disease is produced, that is something worth looking into. And there's definitely going to be a lot of people that are more focused on this aspect, which has more to do with how the virus causes disease and what kind of initial numbers of viral particles will produce disease in the end of the day. And I think those are the people that are probably looking into this, and they'll have much better understanding of what effects it might have.
Okay. But obviously those measures, social distancing, masking, are all things, that's how we deal with this variant, in a sense, is just more rigid or more attention paid to those.
Yes, absolutely. The tool that we've managed to deploy that has been somewhat efficient, and I would say actually considerably efficient at controlling transmission, has been just taking these behavioral actions that get people a bit more distance from each other and a bit less direct contact with each other. So especially if we're thinking about how the virus transmits, of course, these measures may have other effects in the bigger picture, but if we are just focusing on the virus transmitting, I think it's one of the more useful tools that we've managed to come up with.
And I know you're not in the business of making policy recommendations, but the strict lockdown in the UK, many countries are limiting or banning people from Great Britain entering the country, or requiring double testing, things like that. Are those? Do you feel like those are good ways to stop the spread of new variants?
Yes, I've actually had a chance to think about this question, and I think the perspective from the UK is particularly interesting because the UK, as many of you may know or may not know, most of the healthcare system is actually centralized. So the National Health Service, the NHS, provides the healthcare for the majority of the British population. It's actually one of the biggest providers of employment in the United Kingdom. So whenever these measures are taken in the context of the UK, it always has an element of focusing on not overwhelming the NHS. because for once, overwhelmed health systems are less capable of providing support for people who are infected. But at the same time, because that places a big toll and a big pressure on doctors and nurses and healthcare workers in general. So I think that in that context, whenever the UK takes the decision to implement a lockdown, I think given that specific outcome that they're looking for, it is a bit more straightforward to understand why the decisions are being made. And end of the day, I do think I think that lockdowns are they're definitely a hammer as far as measures go. They're not subtle. They're not tailored to really attack the transmission process where it matters. They're broad. They have good effects. They have bad effects. But in the end, when it comes to controlling transmission, I think that we've seen different lines of evidence from different countries that they work. So yes, end of the day, I do believe that that for very specific outcomes like protecting healthcare systems not to be overwhelmed, I think that in those scenarios, the target is clear, and whether a decision is taken or not really depends mostly on balancing out the rest of the effects. So, and of course, it comes down to people that are more knowledgeable than me on everything from economic effects to mental health effects on the population, to compliance, so all of these different factors will play a role. But if you're only asking me, how can we avoid the healthcare system to be overwhelmed with new cases every day, then yes, lockdowns are definitely a valuable tool.
Okay. I have a question from a Tanzanian journalist. I can't quite see the name here, but this is a Tanzanian journalist who is currently in the UK. Tanzania is among other few countries that have not been affected with COVID-19 as much. Now, with the rise of variants in several countries, how should it be prepared, countries like Tanzania, to ensure that it does not get infected or get affected with the new variants? And in other words, Tanzania may be a country that isn't doing this genomic sequencing, so they don't know for sure whether they have this variant. But what kind of recommendations would you have for people in countries where they really don't know if it's spreading?
The entire case of Africa is quite interesting because, the general idea some point in the summer was that it was mostly spared. And by this, I don't mean that they didn't see a burden from COVID, but it was definitely less than some other parts of the world. So it is a very interesting scenario. And I think, I'm not sure if we understand fully why this was the case. Probably based on my own, the research I've been involved in, which might give me a bit of a bias here. But I do think that, for example, the fact that human mobility is not as intense with some countries in the African continent compared to others, that might have played a role. So basically, the more introductions you have of the virus, the more people who are infected coming in, you may see a larger number of cases. But in any case, regardless of what the reason for this lower number of cases in quite a few African countries, I think that the lessons that have been learned from countries that were more hard hit are definitely valuable and worth considering for places who have been fortunate enough to not see big surges in the number of COVID cases at the moment. But having said that, it is about tailoring the tools that are available to your own reality. Locations like New Zealand and Australia have seen relatively well-controlled epidemics. Same has happened for places like Japan, Thailand, Vietnam, South Korea. Asia actually had, from the numbers, it looked like they did have a good mechanism to control the transmission. So I think there are a lot of valuable tools that came from those countries, but it's not about just taking the tools and applying them to your local reality as they come. It is about having a time to think about what kinds of measures would be applicable to different scenarios. I know, for example, with my home country, Ecuador, that it was a very particular situation. The dynamics of how the virus and the epidemic progressed were very particular. And fortunately, eventually it was under control, particularly in one of the largest cities in the coast, which was hit the hardest during the early months. Yeah. But it was a very specific set of circumstances, And that doesn't mean that whatever they did there, A, it was the only reason why the virus was controlled, or B, that the same ideas applied to other places would have the same effect. So it's a bit tricky, but it is a case-by-case scenario at the end of the day.
Right. A question from Barack San Martin. I wanted to ask if this new variant has been detected yet in animals, and do you have any information about the new variant spreading with animal-human interaction?
Oh, that's a very good question. So, to my knowledge, it hasn't been detected in animals yet. To that, I will add that most of the surveillance at the moment is probably being directed towards humans. So that means that we're looking very hard in humans. I'm not sure how hard we're looking in other animals. Now, one thing that is interesting is that some of the mutations that we originally saw in the UK variant, they were seen previously in a specific outbreak in animals, in minks in Denmark.
Mm.
So that doesn't seem to suggest that they are connected in some way, but there may be some mechanisms that led to both variants arising independently that are shared between whatever caused the variants such as the UK variant and variants such as the one that emerged in minks. As far as the second part of the question, which is more on, in general, animal to human transmission and vice versa. So for coronaviruses in general, I think we've had a few years, probably decades, where we have been aware that this is a family of viruses that tends to transmit quite easily between humans and animals, and that's not really a rare thing. So influenza viruses tend to do that. Same thing happens with rabies viruses, which is a classic example. Even things like the Ebola virus is, after all, just a jump from a virus that circulates in animals and into humans. So I think that at this point, there is enough evidence to suggest that there are a few different animal species. Carnivores seem to be particularly vulnerable to being infected with this novel coronavirus from humans, and then the possibility of spillback might be a reality. It doesn't seem to be one of the main drivers of transmission, and it doesn't seem to be something that has led to the emergence of a particular variant or mutation worth looking at, but it is probably part of how the virus transmits. So it's something that, while it may receive a bit less attention because of urgency, I would assume, but it is important to look into how that's behaving at that level of interface between humans and animals.
A question, we talked about vaccines and effectiveness of vaccines. This question from Christina is about safety of vaccines. Is there any data on whether these variants could affect the safety of vaccines as opposed to their effectiveness?
That I'm aware of, I haven't seen anything regarding safety. And of course, I mean, unfortunately, not my area of expertise, but this is something that should be looked at by people who are working most closely with immunology and hopefully the development of vaccines. So yeah, I mean, as any other aspect of a new variant, worth looking into, but unfortunately, I haven't looked at data, okay, nor is it my area of expertise for me to comment on.
From Kunal Saikia, how well prepared do researchers and the global healthcare system need to be to efficiently contain the virus by lessening its transmission when new viruses are emerging rapidly at the same time? I'm not sure if that's new viruses or new variants, but.
So just to double check, so the question was how prepared do researchers need to be, or healthcare?
Well, it's both, the researchers and the global healthcare system. Obviously different answers for each, I would guess.
Yes, because yeah, my first thought was precisely, as far as researchers go, I think that this pandemic has shown that there is plenty of expertise and the will to conduct research on urgent matters such as a novel pathogen, as happened early in 2020 with this current pandemic. I think, of course, the challenge will always be the resources available for that. There's still quite some differences between the kinds of research that can be performed in different countries based on the research on the resources that are available. Now, as far as how prepared healthcare systems need to be, it's definitely going to be something that will need to be a priority for pretty much every healthcare system in the world, given how basically how interconnected the globe, the planet Earth works at this point. There are no countries that are isolated enough so that they don't have to worry about new pathogens in one region eventually reaching their shores. So it's something that it may require the commitment of resources, for example, but I think that it's less about having a very robust investment, as in monetary investment in certain applications, and more about the ideas behind what approaches are going to be used to try and contain outbreaks, local outbreaks. That's going to be important. To emphasize in local variants, of course, the ideal scenario would be performing genomic surveillance, which is definitely, at the moment, it's the cheapest it's been in human history, but it still takes a bit of a learning curve and a bit of infrastructure building. Our experience in Ecuador has been quite interesting because while there was a lot of know-how, actually implementing the protocols, it didn't take that long, but it was its own small learning curve. And then access to samples, to sequence is also a process that's a bit more complicated. So trying to think of that as early as possible, I think, is one of the more interesting ways to be prepared early for potential emergence of things that you can only really reliably detect through sequencing the whole genome, at least at the beginning.
And kind of a follow-up to that, the research that we've been able to do on this novel coronavirus, we're going to see more viruses, obviously, in the future, more pandemics. And are we now, because of the research that you and many others around the globe are doing, are we going to be better prepared for the next one? Or is it going to, each virus is so different that really it's not necessarily going to help us?
Well, they will be different, that's for sure. I do think and hope that there have been plenty of lessons learned with this one. And I think it even goes beyond really just basic science and research questions. I think that for us to really make the most out of this entire and tragic event that has occurred over the last more than, well, getting close to a year to it being officially seen as a pandemic. I think it's about how we've learned about how we think about ways to contain and control a pandemic, how we think about ways to implement diagnostic schemes, things about how we communicate, whether it's ongoing research or certain findings. It's definitely been something, a bit of an eye-opener to see how information has spread, what role social media has had, what role the sitting down on certain political opinions while you're expressing opinions or trying to communicate science. I think that's been something that's we never saw before. And just looking at that unfold over the last year, I think there are lots of important lessons to be learned from the science, and probably as many important lessons to be learned as into how we see a developing pandemic, how we communicate this, and how objective we are actually called to be whenever we're communicating this kind of information in an ongoing situation that will necessarily be uncertain at times.
Mm-hmm. That's a good answer. A question from Maureen Cornio. And this goes back to your what we talked about earlier regarding sort of the research that's currently going on. How can we confirm without any doubt that the transmissibility is increased? Is it by doing animal experiments?
Well, that is going to be an important piece of the puzzle. Coming from a very, very purist, and probably it's going to be an annoying answer, we're never 100% sure about something happening. We get to the point where all evidence aims at the same idea, concept, or model in our minds, and then we can say, okay, beyond all reasonable doubt, so to speak, we believe that this is how it behaves. And I think with science in general, it's interesting because probably one of the best signs that you really understand any natural process is when you can actually replicate it or control it. So if we say, okay, we believe that the close contact between people is driving the transmission of the virus, and we implement lockdowns, and that actually lowers the rate of transmission. If we're seeing things like that over and over again, in a way, and I know it sounds like it's almost like an experiment done on purpose, but in reality, it's just been a consequence of the measures that have been taken. But if we're seeing that there is this consistent pattern of lockdowns and sort of keeping some form of distance between people, that tends to lead to the epidemic curves coming lower. I think those are the kinds of evidence that we really need. The unfortunate thing is, of course, it takes time and data to actually prove those things, but that's sort of kind of where we want to be as far as from a very just scientific perspective, drawing conclusions and expressing our certainty about these conclusions.
Mm-hmm. Okay. And let's see if I can maybe pick out one last question. We're just about at the end. This was a question, I think it's from the same Tanzanian journalist. What measures should Africa, including Tanzania, take to ensure that it does not get affected by these variant viruses? I think the answer is again, those same sort of protocols that countries are using to stop the spread.
Yes, definitely. Take the lessons learned from other countries. Don't expect the exact same measures to be what any country needs. Just look at the toolbox, and based on that, think, okay, what might be applicable here? But definitely overlooking all of the different approaches that the world has had over the last year would be doing a disservice to any public health response.
We are at the end of our discussion, so I'm just going to put one last question out there, which is sort of gets back to the overarching question we had at the beginning, which is about, really how concerned should we be about this? How optimistic do you feel as we're, rolling out vaccines, which is a positive step, and then seeing new variants increasing the transmissibility, which is a negative factor. And as I said earlier, we're kind of in a race. Can we get enough people vaccinated before the new variants just really drastically increase the spread? How optimistic are you that we're going to be able to do that as these new variants are rising?
Well, probably as a rule of thumb, I will tend to be optimistic on our ability to better understand this pathogen or any pathogen and take actions to controlling it better. So again, and going back to my original message, I think the key here is really trying to be as prepared as possible and trying to devote the time and effort and resources to understanding these basic questions. I think at the end of the day, whether it takes a little bit longer or not, we will find ways to deal with the situation. We will adapt both in our behavior, our attitude towards something like a threat as this pathogen, as this virus has been. But yet again, I think end of the day, probably fortune favors the well-prepared would be my approach. So the more that you try to stay one step ahead of the virus and actually do your work before things get out of control, I think that is going to be one of those very important and inciting factors.
Great. Well, thank you very much, Bernardo Gutiérrez. I'm very, very grateful to you for joining us for this webinar. This is a really complicated scientific topic, and I appreciate that you've really been able to explain it in a way that a scientific idiot like myself can understand. So I hope that that many in our audience have a greater understanding now of these variants and what we can expect from them. So thank you again. For those of you who speak Spanish, Bernardo Gutiérrez will be doing a Spanish webinar as well on Thursday. So check our forum in Spanish for details on that, and please register for that webinar if you speak Spanish. I want to remind everyone that this webinar is recorded and we will be putting it up on ICFJ's YouTube channel within 24 hours. And we ask you to please fill out an audience feedback survey that we'll be sending all of you who registered after the webinar so we can get your feedback. It's very important for us to know what kinds of webinars you find most useful, what works for you, what doesn't. So we really appreciate those of you who have registered to fill out that survey. Remind you again to visit our ijnet.org website, where you can get lots and lots of resources in 8 languages on covering the pandemic and dealing with disinformation and many, many other important aspects of the story of our lifetimes. So again, thank you, Bernardo Gutiérrez. Thank you to all of you for joining us for this webinar. Please visit our Facebook page and join us for the next webinar. We'll be doing one a week as we continue through this pandemic. And stay safe, everyone. Thanks again.
Thank you very much. Thanks, everyone.